Technobotany by Zura. Today we'll cover Western blotting, a fundamental technique for detecting specific proteins in complex samples. We'll walk through the introduction, underlying principles, step-by-step methods, and key applications. What is Western blotting? Western blotting is a powerful laboratory technique used to detect and measure specific proteins within a complex mixture. It works by first separating proteins based on their size using electrophoresis, and then identifying the target protein through antibody-based detection. The name Western blot was coined as a playful nod to other blotting methods, like Southern blotting for DNA and Northern blotting for RNA. Today, Western blotting is a cornerstone in biological research, clinical diagnostics, and the biotech industry, helping scientists uncover the molecular details of life. Steps in Western blotting technique. Western blotting follows a multi-step process to identify specific proteins. It begins with sample preparation, in which proteins are extracted from cells or tissues. In Step 2, gel electrophoresis (SDS page) is performed, where proteins are denatured and separated based on their molecular weight. In Step 3, the separated proteins are transferred from the gel onto a stable membrane. Step 4 involves blocking and antibody incubation, during which nonspecific binding is prevented and specific binding is enhanced. Next, in Step 5, protein detection and visualization are performed, where the target protein is identified and made visible for analysis. In the following slides, we will discuss each step in more detail. Sample preparation. Effective sample preparation is essential for reliable Western blot results. The process begins with cell lysis, using buffers like RIPA or NP40, combined with protease and phosphatase inhibitors to preserve protein integrity. Next, the protein concentration is measured using assays such as BCA or Bradford to ensure equal loading. Gel electrophoresis. Gel electrophoresis, specifically SDS page, is the method used to separate proteins based on their molecular weight. Proteins are first denatured in a loading buffer containing SDS and reducing agents like beta-mercaptoethanol, giving them a uniform negative charge. This ensures that their migration through the polyacrylamide gel is determined solely by size. The gel percentage, typically between 8 and 12%, is selected depending on the size of the target protein. During the run, conditions like buffer composition, voltage, and temperature are carefully controlled to maintain consistency. Molecular weight markers are included alongside samples to help identify the size of the separated proteins. After electrophoresis, proteins are transferred from the gel onto a membrane, a process known as blotting. This can be done using wet, semi-dry, or dry transfer methods, each with its own advantages depending on the experiment. The membrane itself is typically made of PVDF, known for its strong protein binding capacity, or nitrocellulose, which offers ease of handling. Transfer efficiency is influenced by factors like pore size, uniformity, and duration of the transfer. To confirm successful protein transfer, researchers often use Ponceau S staining or other reversible dyes that provide a quick visual check before proceeding to antibody detection. Blocking and antibody incubation. Once proteins are transferred to the membrane, blocking is the first essential step to prevent nonspecific antibody binding. This is typically done using a solution like 5% BSA or non-fat dry milk in TBST. After blocking, the membrane is incubated with a primary antibody that specifically binds to the target protein, with careful attention to dilution, buffer composition, and incubation time. Next comes the secondary antibody, which is species-specific and conjugated to a detection label such as horseradish peroxidase or a fluorophore. While protocols can vary, a common timeline includes 1 hour for blocking, overnight incubation with the primary antibody, and 1 hour for the secondary. Detection methods. Western blot detection relies on several methods to visualize the presence of target proteins. Chemiluminescent detection is the most common, using horseradish peroxidase and a substrate to produce light, which is captured on film or with a digital imaging system. Fluorescent detection offers quantitative analysis and multiplexing capabilities, allowing multiple proteins to be detected simultaneously. Colorimetric detection, though less common, uses alternative substrates that produce a visible color change. Radioactive detection uses radioisotope-labeled antibodies to bind target proteins, allowing their visualization through autoradiography based on emitted radiation. When choosing a detection method, it's important to consider factors like dynamic range, exposure time, and signal linearity to ensure accurate and reproducible results. Image acquisition and analysis. Once the signal is generated, capturing high-quality images is crucial, with careful attention to exposure settings to avoid saturation or underexposure. Quantification is typically done using densitometry, which measures band intensity to assess protein levels. To ensure accuracy, results are normalized either to loading controls, like housekeeping proteins, or to total protein using methods such as Ponceau staining or stain-free imaging. Finally, robust analysis includes both technical and biological replicates, allowing researchers to distinguish experimental variation from true biological differences and apply appropriate statistical methods. Common loading controls. Loading controls are essential for accurate interpretation of Western blot data. Commonly used housekeeping proteins include beta-actin, GAPDH, and tubulin, though it's important to recognize that their expression can vary under certain experimental conditions, so validation is key. As an alternative, total protein staining methods like stain-free imaging or Ponceau staining offer a broader view of protein loading across lanes. Regardless of the approach, confirming the stability of your loading control under your specific experimental setup is critical to ensure reliable normalization and meaningful comparisons. Variants and related techniques. Western blotting has evolved into several variant methods that offer unique advantages. Dot blot simplifies the process by spotting samples directly onto a membrane, while capillary Western, also known as WES, automates the workflow for higher throughput and reproducibility. Quantitative Western blotting enhances precision, allowing for more accurate protein measurement. Beyond Western blotting, related techniques like ELISA, immunoprecipitation, and mass spectrometry are widely used in proteomics. Each method has its strengths and limitations. For example, ELISA is ideal for high-throughput quantification, while mass spectrometry provides detailed protein identification. Choosing the right approach depends on your experimental goals, sample type, and required sensitivity. Applications of Western blotting. Western blotting is a cornerstone technique with wide-ranging applications in science and medicine. It is commonly used for the identification of specific protein targets, analysis of RNA protein, DNA protein and protein-protein interactions, and detection of post-translational modifications. It also aids in determining subcellular localization, antibody characterization, detection of protein isoforms, and epitope mapping. Furthermore, Western blotting plays an important role in the diagnosis of autoimmune disorders as well as infectious and non-infectious diseases. I hope you enjoyed this video.